Hi everyone. We are going to learn today the very introductory part of what's called regulation of genes. When I say regulation of genes, it means the activity of the gene itself. You know, gene is a small segment of DNA. It may be, when I say small segment, it may be 10 nucleotide sequence, maybe 100 nucleotide sequence, maybe 10,000, maybe more than that. But it's a small segment of DNA that will code for one polypeptide chain having amino acids. And such polypeptide chains may form a protein. And therefore, a protein is a polypeptide chain made up of many nucleotide bases in a particular way. That is done by the gene. And regulation of genes means controlling of the expression of the gene, controlling of the gene itself. You know, sometimes in a disease like cancer, cell division and cell growth cannot be controlled. Uncontrolled division of cell will go on. Then genes have lost their function in that condition. Therefore, the living system always takes care to control the expression of the genes in the form of proteins. Now, proteins are required for structural part. Every living cell is made up of a large number of structural proteins, and they are required for function or functional proteins, especially enzymes. All enzymes are proteins, and therefore that's also required. Each one of these things are very different. You know, there are different types of proteins in our body. All these are made by the genes, and also some special activities. It may be something like controlling the activity of a virus attack. It's a special activity. Some regulation will have to happen, and when the people mature at some time, and they get the mustaches, they get hair all over the body, the voice will change. All this will happen. All these are controlled and regulated by genes. And then catalytic activities. We take food every day, every time when we are hungry or not hungry, and that food has to be broken down, and that broken down food has to be burnt. That's catabolism. For that, enzymes are required. And when once the digestion takes place, the molecules are produced. These molecules will have to go back to form the structures, maybe carbohydrate structure or maybe protein structure. Then also we require enzymes. So that means every activity of the living cell, which takes place in multitudes of functions every second as long as we are living, and all these are controlled by genes, and that expression of the genes will have to be regulated and also controlled. If there is no control, there will be catastrophic activities in the system. So at 4 different levels, the gene expression is controlled. Number 1, at the transcription level, that is when the DNA strand opens, one strand called sense strand remains as it is, and the other one, antisense strand, now becomes a template. On that, the genetic information is copied, and that's called the transcription. So at that level also there is a control, because transcription will not happen the entire length of the DNA. It will be made only at certain points, so there is a control there. And then gene splicing. When the transcription is made, it's made into a very long polypeptide chain with degenerate nature of the nucleotides, or there will be redundant nucleotides which are not useful. They will have to be cut and made into a regular gene. And then also that is second level of controlling. And third level is transporting of mRNA. And once the mRNA is made on the DNA strand, and that mRNA has to be transported into living inside the cell from nucleus to the cytosol, where ribosomes are there. So then also there will be a migration. You know, these are all, we are talking in terms of the angstrom units, 1 billionth of a centimeter or millimeter. That is the magnitude of the smallness of these materials. So at that time they have to be controlled, and then comes translation level. That means when once the mRNA and ribosomes and tRNA come together, they will collect the amino acids and they make the proteins. There also it has to be controlled. So at 4 levels gene regulation takes place. Again I will tell, the first level is the most important level, the transcription level on the DNA, and the gene splicing level, that also happens in the nucleus, and then the transport of the mRNA that is made on the template DNA comes to cytosol. They bind to the ribosomes, and then there also there is a control which area this has to be attached, etc. will be guided by proteins, and then finally at the translation level it controls. These are the 4 levels at the cellular level, at the level of the cell. But if you take the entire the system as a whole, even inside the cell, there will be metabolic activities. That means some will be destructive activity, like breaking down the molecules. For example, glucose will be broken down into carbon like that, and then ultimately energy is released from that. That's the catabolic activity. And anabolic also takes place. That means from the small segments of these sugars, they will be added to become polysaccharide, and that becomes a sugar again. Similarly, amino acids will form, amino acids will become the protein. That means both these activities will be happening at the same time. And also at the environment of the cell itself, where all these things are happening. What kind of things, if any new structure has gone in, the chemical nature of the cell has changed, so then also there will be regulation. That means the genetic regulation of the production of the proteins especially will take place at different levels by different factors, and all that is termed as gene regulation. It may be expression into a definite type of a protein, or it may be a suppression. Something may be reduced also. That will also happen. This type of work expressed right from the time of Crick and Watson, when they discovered about the DNA, but more so about the people when they started working on E. coli bacteria, especially Jacob and Monod worked on one particular metabolic activity where how lactose is utilized, and then they identified what's called lac-operon concept they give. Operon refers to a functional unit where many related genes come together. Many segments of DNA come together which work as a gene, and they will be controlled by 2 important parts. One is called the promoter, other is called the operator, and that becomes a operon. There are hundreds, and it is said that E. coli has nearly 500 to 7,000 operons in E. coli, a small bacterial cell which is microscopic in nature. Then you can understand how many more operons are there in the human beings, where we have millions of genes in our body. So how an operon functions, a model has been given by Jacob and Monod, and that we will study in the next session. Thank you.